All right, everyone, welcome back. Today, we're going to dive into one of the most incredible stories in all of human biology. We're talking about the life of an oocyte, the female egg cell. This is the process we call oogenesis, and trust me, it is an absolutely fascinating journey. Okay, so here's our game plan. We'll kick things off with a quick overview, then we'll rewind all the way back to before birth to lay the foundation. After that, we'll see what kicks into gear at puberty, untangle the very unique process of meiosis, cover the final steps to ovulation, and then we'll wrap it all up with a nice, clean summary of the key takeaways. Let's get into it. All right, first things first. Before we get into the weeds, we need to make sure we're all on the same page. So what exactly is oogenesis? So here's the bottom line. Oogenesis is not just one thing. It's the entire long and complex process of transformation. It's how a very, very early stage germ cell, what we call an oogonium, develops and differentiates all the way into a mature oocyte, or egg, that's ready to be fertilized. Now here's where things get really interesting and quite different from what happens in males. The story of every single oocyte a person will ever have, well, it actually begins long before they are even born. Just look at the drama unfolding on this timeline, all within the fetal ovary. You had this massive explosion of cell division, followed by a peak, and then a huge programmed die-off called atresia. So by the time of birth, every potential egg for that person's entire reproductive life is already created and has hit the pause button in the very first stage of meiosis. And get this. At its absolute peak, around the 5th month of pregnancy, that fetal ovary is packed with about 7 million of these germ cells. That's the absolute maximum number it will ever have. It's just a staggering amount. But from that incredible peak of 7 million, atresia, that programmed cell death, kicks in hard. So by birth, the number has plummeted to somewhere between 600,000 and 800,000. That's a reduction of over 90%, and this is all before life outside the womb has even begun. So this is what we're left with at birth. Each of those surviving primary oocytes is now wrapped in a single protective layer of flat cells. This whole package, the arrested oocyte plus its little cellular wrapper, is what we call a primordial follicle. This is the basic, dormant unit sitting in the ovary, just waiting. Okay, now let's hit the fast forward button. We jump ahead more than a decade to puberty. This is when, each month, a small group of those sleeping primordial follicles gets the signal to wake up. Their support crew finally gets the call to get to work. You can see this incredible evolution here. The follicle goes from that simple resting state to a primary follicle where the support cells plump up and start dividing. Then it becomes a secondary follicle when a fluid-filled cavity, the antrum, starts to form. This antrum is like a nutrient-rich soup for the oocyte. This all leads up to the final stage, the huge, mature Graafian follicle, which is basically a complex little temporary organ primed and ready for ovulation. And that support crew is way more than just packing material. The follicular cells transform into granulosa cells, which build that critical protective shell, the zona pellucida. And surrounding them, the ovarian tissue organizes into 2 more layers, the theca interna, which is a little hormone factory, and the tough outer theca externa. They are actively controlling everything. Now let's talk numbers again, because they're just wild. By the time puberty starts, atresia has continued, and we're down to about 40,000 oocytes. Each month, a cohort of maybe 15 to 20 gets recruited to mature, but usually only one single oocyte wins the race and gets ovulated. Over a lifetime, that means fewer than 500 will ever even get a chance at fertilization. The system is incredibly selective. Okay, let's get into what is arguably one of the most unique and clinically important parts of oogenesis. We have to talk about its weird start and stop division process. You absolutely have to get your head around these two great pauses. So you got to ask yourself, right? Why is it like this? Why is it so different from the continuous factory line process we see in males? Well, this start-stop mechanism has some really profound consequences for reproductive health. And here they are, the 2 big pause points. Pause number 1 happens right at birth, in prophase 1. The oocyte is basically frozen in time for years, sometimes decades, held in check by an inhibitor from those follicular cells. Then the LH surge at puberty finally breaks that first arrest. The oocyte rushes to finish meiosis 1 and start meiosis 2, only to hit a second pause button, this time in metaphase 2, right before ovulation. And this second arrest is only released by one thing: fertilization by a sperm. I mean, just let that sink in for a second. An oocyte that is ovulated when someone is 40 years old has been sitting in that first meiotic pause for over 4 decades. Now, clinically, this is huge. It helps explain why the risk of chromosomal problems, like trisomies, goes up with maternal age. The cellular machinery that has to hold those chromosomes together perfectly can just start to wear out over that much time. So let's talk about that trigger. What's the signal that finally breaks that decades-long pause and pushes the oocyte towards the finish line of ovulation? It's all about the LH surge. In the middle of the menstrual cycle, there's this huge spike in luteinizing hormone, or LH. This is the big green light. This hormonal flood overcomes those inhibitors from the follicular cells and basically forces that primary oocyte to wake up and finally complete its first meiotic division. But here's the thing, this division is completely lopsided. One cell, the secondary oocyte, gets to keep pretty much everything, all the cytoplasm, all the nutrients, all the organelles. The other cell, called the first polar body, is basically just a little garbage bag for the extra set of chromosomes. This is absolutely critical because it makes sure the future zygote has a massive stockpile of resources to survive, and it ensures that all of our mitochondria, our cellular powerhouses, are inherited from our mothers. So after it's been ovulated, the secondary oocyte is at its final checkpoint, paused again in metaphase 2, and now the clock is ticking. It has a very brief window, only about 24 hours, to be fertilized. If a sperm doesn't show up in time to signal it to complete meiosis 2, the oocyte just degenerates. The journey ends there. Okay, I know that was a ton of information, so let's zoom out and pull it all together into a nice, high-yield summary that's going to be perfect for when you're studying. This table right here is your best friend for revision. It perfectly connects what the follicle looks like on the outside to the meiotic stage of the oocyte that's hiding inside. And pay attention to that little note at the bottom. The oocyte stays as a primary oocyte all the way through the secondary follicle stage. It only completes meiosis 1 and becomes a secondary oocyte right at the very end, thanks to that LH surge. So look, if you only remember a few things from today, make it these 4 points. 1, it all starts before birth with a huge loss of cells. 2, meiosis is defined by 2 major pauses. 3, those follicular cells are the active support crew running the whole show. And 4, the whole process is incredibly inefficient by the numbers, with very few oocytes ever getting a shot at fertilization. And that brings us to a final question, just some food for thought. When you think about that decades-long dormancy, you have to wonder, what amazing cellular machinery does the oocyte have to protect its DNA all that time? And what happens when those protective systems start to get old? This really connects all the basic science we just covered to the real-world clinical problems of infertility and genetic disorders. Thanks so much for watching.